Fantastic. Good morning, everybody. Thank you so much for having me today for this presentation on creatine in pregnancy and perinatal brain injury. So my only disclosure is that I do sit on the wonderful Creatine for Health Scientific Advisory Board of the ELSCHEM group. So putting together my presentation today, I couldn't help but reflect on the last time I was here and we were all in person back in 2015, so 10 years ago, in beautiful Laufen, which is not too far away from here. I had literally just submitted my PhD, focusing on maternal creatine supplementation in our cute little spiny mice and looking at whether it was useful to protect the infant from a condition known as acute kidney injury. Now, as a part of that study, we just did a little look at how the mums were responding to the creatine supplementation, just because we thought it would be fun and the animals were there. And we noticed something really interesting, and it wasn't from our creatine supplemented mice, but it was actually just from our control mice as we tracked different measures of creatine synthesis, creatine breakdown across pregnancy. And we saw that there appeared to be maternal adaptations to creatine homeostasis just being driven by the fact that the mouse was pregnant. And so since then, we've made it a focus of our research to really try and hone in on what these changes are to creatine homeostasis during pregnancy. Are they important? And do they have any association with pregnancy complications? And I'm not sure whether it was just because I've been in the mix, but it feels like in that 10-year bracket that creatine in pregnancy is being discussed a lot more broadly, not just by ourselves, but by other groups around the world. So I did do a quick search in Scopus of creatine in pregnancy, and indeed we've seen this upward trajectory from around that 2015 meeting. Still small numbers, it is a niche topic, but it's definitely gaining some momentum. And these increased publications are not just the hypothesis-driven papers that our group and others have put out, but also the vergence of bioinformatics and large metabolomic studies. Pretty much every paper that comes out doing a metabolomic study on a pregnancy complication, creatine is usually flagged in the data as being significantly different between the controls and the cases. But interestingly, it's very rarely focused on within the papers. It's usually just a little byline to say, and we found that creatine was significantly changed. Creatine's important for energy, the end. And so I think it's really interesting that data is coming through, and it's an important thing for us as the creatine community when we see these papers to actually try and educate the audience to say that this is actually a compound or a molecule that we should be focusing on in a little bit more detail. But indeed, there is strong emerging evidence that alterations to creatine homeostasis do occur with the natural progression of the healthy female reproductive cycle and pregnancy. So in terms of my presentation for today, I would like to go through the latest in this space. So we'll look at an overview of creatine metabolism in key reproductive tissues. We will look at changes in creatine metabolism associated with pregnancy complications to get at this question of could maternal or neonatal creatine supplementation be used to improve outcomes for mother and baby. We will also look at some of the maternal characteristics that we've identified as being associated as changing the availability of creatine during pregnancy. And today I'll particularly focus on some of our findings associated with diet to look at this key question of whether dietary creatine may actually become conditionally essential nutrient during pregnancy. So we'll start at the beginning, and the beginning for pregnancy is our beautiful uterus, which is made up of two key layers. That's the endometrial layer. This is the layer that grows and sheds across the menstrual cycle, as Sam beautifully described for us yesterday. And then there's the underlying myometrial layer, which is the smooth muscle layer of the uterus, which is important for maintaining the stability of the pregnancy, but also, of course, producing those strong contractions during labour to deliver the baby. And so we conducted a study where we accessed banked, fixed endometrial samples from women across the menstrual cycle. So we had 5 different stages of the menstrual cycle where we had tissue collected, and we were able to look at the creatine transporter and synthesizing enzymes, the CKBB and ubiquitous mitochondrial creatine kinase, and then also in some of our samples look at creatine metabolites, so creatine and phosphocreatine, and also the amino acids arginine, glycine and methionine. The take-home message from this paper was that we saw the expression of the creatine transporter and creatine synthesizing enzymes for the first time within the endometrial samples across the menstrual cycle, but with the expression being highest in the early secretory phase of the cycle. So this is when the endometrial tissue is starting to build up in preparation for the potential implantation of a blastocyst to develop an embryo and then eventually a baby. Now, the interesting thing about this data and this biobank that we got access to was that we had samples from women who had had babies, but also samples from women who were experiencing primary infertility. And when we looked at the expression of the creatine synthesizing enzymes, the creatine transporter and creatine kinase between the 2 populations, we saw an interesting finding with the creatine transporter, particularly in the proliferative phase, where there was a higher level of expression of the creatine transporter in the women suffering from primary infertility. Now, going back to this original slide, the proliferative phase is right down here, just when the tissue is gearing up for this massive period of growth in preparation for the potential implantation of an embryo. So something is happening in these tissues around that time to upregulate creatine transporter expression when there are fertility complications. Now, if we look at a little bit more detail at the human uterus during pregnancy, I say that this data is new, but there's actually been studies out for a really long time that have looked at different components of creatine metabolism in the uterus. And some take-home messages is that creatine is present and at higher levels at term pregnancy compared to non-pregnant state. At term pregnancy, the uterine muscle has the capacity to endogenously synthesize creatine. And we see creatine kinase expression increase across the gestational period to being 3 times higher than the start of pregnancy by the time we get to term. And so that's what's happening in the uterus. Definitely evidence there to suggest that creatine is being utilised as an energy source for this tissue across the menstrual cycle and also with pregnancy. Now moving on to the placenta, which is definitely my most favourite organ. Obviously, a pregnancy is all about having a beautiful, healthy baby delivered at the end. That's never going to happen if the placenta isn't growing and functioning properly. This is an organ that needs to simultaneously grow while supporting the growth of the baby. It's important for gas and nutrient exchange, but also for the synthesis of a lot of hormones and proteins that are key to the development of the fetus. So we should never dismiss the placenta, even though it's a particularly ugly looking thing. And so back in 2017, we did a study where we took healthy placental samples from cesarean delivered deliveries. So these were samples that had not gone through the strains of labour. And we just looked to see what the expression was like of AGAT and GAMT, the creatine transporter, and creatine levels. And we found that indeed the placental tissue did express AGAT and GAMT, so the synthesizing enzymes of creatine. In an in vitro experiment, if we gave placental homogenates the building blocks of creatine being arginine and glycine and then methionine, in 2 steps we could actually get the tissue to produce first guanidinoyl acetate or GAA, the precursor to creatine, and then methylate creatine. So this study suggested that at least at term, the placenta had the capacity to synthesize creatine. We also noted the high expression of the creatine transporter, and that was in the syncytial trophoblast layer of the placenta. And so why that is significant is that this is the layer that sits just between the two circulations. So it sits between the maternal circulation and the fetal circulation. It's where we see the vast majority of transport between the two. So whether the creatine transporter is there to support the energy demands of the syncytial trophoblasts, which are high unto themselves, or whether it's there for transport of creatine from the maternal to the fetal circulation, we're not sure at this stage, but it is there and likely to be doing something important. So this is where it started to get interesting. We were lucky enough to get our hands on some samples of placentae from pregnancies that had been compromised. And so on the left here I've got, on the left click happy. We've got samples from placentae of pregnancies that ended with fetal growth restriction, and then on the right with preeclampsia. So both conditions that are associated with chronic nutrient and oxygen restriction to the fetus, highly inflammatory environments, really not a great environment for good energy homeostasis. And I'll admit that when we first set out to do this study, I had hypothesized that we would actually see a decline in creatine concentration within the placentas of FGR and PE pregnancies, because we'd just shown that the placenta could synthesize creatine. These placentas are not healthy. I thought that they would have potential issues with being able to synthesize creatine and thus would have a lower creatine content. But like most things in life, I was very wrong. We actually found a marked increase in creatine concentration in the FGR and PE placentas compared to controls. So in our FGR placentas at delivery, the creatine content was 43% higher than controls, and in the pre-eclamptic placentas, it was 38% higher than controls. But if we consider this in the context of the uterine data that I just saw, we're seeing a similar pattern here of increases in creatine, either production or delivery to the tissues, as being a potential mechanism or adaptation when the pregnancy is not going the way that it should. And now I'm just going to show some data that we were generated through our creatine and pregnancy outcomes study to support this idea and this hypothesis that we're building. So this study was conducted with my colleagues at Monash Health. So again, Dr. Deb de Guingand, who is a midwife, and Professor Kirsten Palmer, who is an obstetrician. We bandied together and decided to do a prospective cohort study of healthy pregnancies to look at creatine alterations across pregnancy and whether there were any associations of creatine with pregnancy outcomes, maternal characteristics, etc. So we had 282 wonderful participants in this study who consented to have blood and urines taken 5 times throughout their pregnancy. We collected a 24-hour food recall at the same time that we collected the blood and urine samples at those antenatal time points. We then attended their deliveries and collected cord blood samples, so both arterial and venous, so the blood that's going to the fetus and the blood that's coming back out from the fetus, a maternal blood and urine sample at the time of delivery, and of course a placental sample. And so the data that I wanted to just show you in this part of the talk is some data connected to creatine levels around the time of delivery of the fetus. And so the first interesting thing to note was a positive association between venous cord plasma creatine concentrations. So this is the blood that is travelling from the placenta to the fetus. It's a little bit confusing because we normally think of venous as being our deoxygenated blood, the depleted blood going back up through the heart and the lungs to come back. With the placenta, the circulations are switched. So we actually have our venous blood is our highly oxygenated blood with all of the nutrients travelling towards the fetus. And so what we saw was that with the length of the first stage of labour, the level of creatine being delivered to the fetus increased. Now, the first stage of labour is that stage where we have ramping up contractions of the uterus, the cervix is dilating, we're getting ready for the delivery. So this is the stage that is the most strenuous, that hurts the most, and goes for the most significant amount of time with labour. And we see that the longer it went, the more plasma creatine that was being delivered to the fetus via the venous circulation. We also saw an association between creatine levels in the cord bloods and the lactate measures of the bloods after delivery of the fetus. So cord lactates are used clinically as an indicator of how well the fetus and the baby did throughout the labour process. So the more hypoxic the labour, the higher the levels of lactate. If the lactates are particularly high, the baby will be monitored post-delivery in case there's any further complications. And again, we saw this positive association in both venous cord creatine concentrations with lactate and also arterial cord creatine concentrations and lactate. So this is both the blood being delivered to the fetus and then returning from the fetus to the placenta. The higher the lactates at the end of labour, the higher the creatine concentrations within those circulations. And now finally, some data on this point that's come from some of our animal studies, so our instrumented fetal sheep studies. I'll go into this in a lot more detail in a few more slides' time in terms of the model and why we use it. But the advantageous thing about using animal models is obviously we can collect fetal tissues and do some invasive measures that we wouldn't be able to do in samples collected from mums and babies. And I just wanted to point out some of the data that was the creatine concentrations in the brain, fetal brain, 72 hours after a severe hypoxic insult. So we've got multiple regions of the brain here. As was mentioned yesterday, the concentrations of creatine in the regions varies. So anyone doing any brain research, just to reiterate the points from yesterday, it's really important to be region specific. I want you just to focus on our saline controls for a second. Ignore the creatine supplemented animals. We actually saw this phenomenon where 72 hours after the insult, we saw increases in our saline hypoxic animals in some of the regions, which actually reached significance in the striatum, which is a key white matter region. So there seems to be, again, an adaptation with a hypoxic insult to increase creatine concentrations within some regions of the fetal brain, particularly key white matter regions. I was actually really annoyed when I first saw this data because it made our primary objective of this study, which was to compare how well our creatine supplemented animals did compared to our controls, really challenging because at the time in which we collected some of the tissues, the level of creatine in our control animals was actually as high as the creatine in our supplemented animals. So that's science though, isn't it? We worked around that. So taking all these little bits and pieces together, so some information from the uterus, some information from the placenta, some information from the fetus, from multiple different species, we see a likely hypoxia-driven mechanism to increase creatine availability to the fetus when there are complications. And so that begs the question of, well, could we use a supplementary creatine earlier on in the pregnancy before we have these hypoxic events, which sometimes, particularly those that occur in labour, are really challenging to be able to predict that they're going to happen. So there's not anything you can really do to prevent them. But if we had creatine supplemented by the mum, increasing creatine levels to the fetus before a hypoxic event, could that additional creatine help maintain ATP homeostasis throughout that period of oxygen deprivation and in the hours afterwards to help prevent injury to the fetus that's associated with hypoxia ischemia? And so really this is the brainchild of my PhD supervisor and mentor, Professor David Walker. David's amazing fetal physiologist who stumbled across some of those early brain HI studies that were done in rats that Professor Wallerman mentioned yesterday. And he saw a lot of parallels with the way in which brain injury manifests in fetuses and newborns following a hypoxic insult. And so he really came up with this hypothesis of whether we could use creatine to buffer this injury cascade that we see occur when there's a hypoxic insult around the time of birth. And when he was looking up who to potentially speak to about creatine homeostasis, he stumbled across my other mentor and dear friend, Professor Rod Snow from Deakin University in Melbourne. At the time, Rod was a really prominent researcher in the creatine space, doing a lot of work on the creatine transporter, and serendipitously, he was 15 minutes down the road from where David was at Monash, where he was starting to work up these ideas. So we've been all working together for quite some time now on this hypothesis and this work, and it's my absolute pleasure to present it on behalf of both Professor Walker and Professor Snow. And so this is just a schematic again to show the different areas in which creatine might actually be beneficial in preventing the energy collapse and injury cascade that we see with hypoxic ischemic injury to the fetus, particularly in the brain. So we have primary energy failure following the hypoxic insult, which obviously depletes ATP and phosphocreatine, leading to this manifestation of oxidative stress, excitotoxicity, mitochondrial dysfunction, and so on, going down into a latent phase where we then see cell death and increased inflammation. We have a secondary energy failure that occurs after these events. So this is usually around 24 hours after the hypoxic insult, which manifests clinically as seizures, and then eventually we see adverse neurological outcomes in these poor babies. And so creatine really has the potential to target this cascade at many different areas. And so we started these studies using our spiny mouse, which I mentioned earlier. This is a wonderful little rodent to use for pregnancy studies because it actually produces precocial young as compared to rats and mice that a lot of people use as laboratory rodents. I'm not sure if you've ever seen a newborn rat or mouse, but they're tiny little jelly bean kind of things, eyes and ears closed, not anywhere near the developmental level as a human fetus. This is a spiny mouse on day one. So they pop out looking like this, eyes and ears open, organogenesis mostly complete by the time of birth, and importantly, some key brain maturation measures such as myelination occur in utero as what happens with us. And so we think this is a wonderful little mouse to do pregnancy studies because of those parallels. So the initial studies were conducted with the mums getting a dietary creatine supplement, so 5% weight per volume of their diet for 20 days of their pregnancy, which is half of their pregnancy, so the second half of their gestation. The mouse is also really helpful because their gestation length is 39 to 40 days, which is actually really a nice parallel with human pregnancy being 39 to 40 weeks. So just replace days with weeks, and we're thinking about kind of what's happening in the human context. So when we caused a severe hypoxic insult at the time of delivery to these pups, what we found was that the pups being born of the creatine supplemented animals, we had a higher survival rate. The pups were protected against brain pathology associated with inflammation, necrosis, and apoptosis. So this was looking at key proteins within tissues, sections of the brain. We found some markers of protection there. We also looked and presented some work to show that the kidneys were protected, and that's important because acute kidney injury in the neonate is a risk factor for poor neurological outcomes as well. If the kidneys aren't functioning well to better facilitate acid-base homeostasis in the fetus, then you get a build-up of ROS and that's detrimental to the brain. So if we can protect the kidney as well, it's good for the newborn overall. And we also saw some benefits of the creatine for the structure and function of the diaphragm and skeletal muscle. So some really nice preliminary data, well, not preliminary, but initial data to suggest that this hypothesis of creatine being useful in pregnancy might actually be worth exploring further. And just one more study that has literally just been published from this set of spiny mouse experiments was looking at the behaviour of the offspring all the way out to 72 days postnatal age. This was done by our amazing postdoc, Nhi Tran. And we found that there weren't a huge number of behavioural changes in our asphyxic pups, but of the few subtle changes that we did see, creatine was able to reduce specifically a hypoxia-induced anxiety-like behaviour in our adolescent-aged offspring. And so we were conducting these studies at the same time as there have been a few other hypoxic ischemic insult studies done in rats and rabbits. And so looking at all of that data together through a systematic review a couple of years ago, it was all looking promising that we should continue to explore this idea of creatine supplements to protect the fetus and newborn against hypoxic ischemic injury. But whilst the rodent studies are really helpful for generating data fast and for looking at certain pathways and different elements of injury and prevention, they don't allow us to get the whole picture of what's happening physiologically to the fetus. And I come from a department of physiologists. We love knowing what's happening in real time with heart rate, blood pressure, oxygen saturations, that kind of thing. And so we then turned to the fetal sheep model to more explore the physiological changes associated with creatine and just to get some more robust data around what's happening and potentially getting at some mechanisms. So the sheep are a wonderful model for these kinds of studies because a fetal lamb is actually a really similar size to the developing baby. In terms of development and maturation, it's on a similar trajectory to a human baby. It's a little bit accelerated, so they're born a little bit more mature than we are, but it's not too bad in terms of a like-for-like comparison. And we can also do surgeries on the pregnant sheep and the lamb fetuses to insert catheters, probes, you name it, to measure a whole bunch of different factors across the pregnancy and then before we collect tissues and actually look at outcomes. So I'd like to present two studies today, again being driven by Dr. Nhi Tran, who it would have been lovely for her to be here today to present this work because she's really been the cornerstone of getting this beautiful set of data. She would have loved to have been here, but she herself had a little girl just 5 weeks ago. So I think she's at home listening online. So hello, Nhi, if you're there. I'm sure I'll get some notes from her, but hopefully I do her justice with presenting this data today. So we have two separate studies that I'm going to touch on. The first one was really looking at that primary stage of energy failure. We inserted a microdialysis probe into the brain and it was infused with terephthalic acid so that we could pick up changes in reactive oxygen species across the duration of the experiment and with the hypoxic insult. And then the second study, we focused on electrographic activity, particularly seizure burden within the fetuses. So a bunch of probes went in for us to be able to measure those outcomes. After surgery, the animals are allowed to recover before they get a direct fetal infusion of either creatine or saline. Now, it's very early, but hopefully you're going, direct fetal infusion? Aren't we talking about a maternal supplementation? What's going on here? This is a workaround for the sheep because they're herbivores and they eat grass and they don't consume any creatine through their diet. And based on some very expensive initial studies that we did, it doesn't seem to cross the sheep gut particularly well, and it doesn't cross the placenta. So whilst we know that we can get creatine to the human fetus across the human placenta, and we can in other animals that are omnivores, the sheep, not so lucky. So we did take the approach of giving a direct fetal creatine infusion to the sheep. After a period of time, we conducted an umbilical cord occlusion to induce a hypoxic insult, a severe hypoxic insult to the near-term fetus. The two hypoxic insults were slightly different between study 1 and study 2. Study 1 was a set period of time of hypoxia. Study 2, we aimed for a particular drop in blood pressure. Both were severe hypoxic insults designed to induce brain injury. We then let the fetuses recover for 72 hours before collecting tissues. And over here we just have some data showing the differences in plasma creatine concentration in the fetuses across the supplementation period, just to show that our direct creatine infusion does increase the plasma creatine levels of the fetus before we do the UCO or the hypoxic insult 10 days after the supplementation. And so the first set of studies, as I said, was really focused on looking at that primary energy failure. And we did this looking at, as I said, microdialysis to look at ROS development in real time and also to collect key metabolites from the brain. So focusing on that development of reactive oxygen species to start with, using terephthalic acid as to capture the ROS production, and then measuring the difference in terephthalic acid as a marker of ROS. We can see that our creatine supplemented animals here in the blue, we did not see as high an increase in ROS production in really the first 24 hours after the hypoxic insult compared to controls. As you can probably tell from the graph, it was somewhat cyclic, which fits in with those that paradigm of the energy failure cascade and an initial primary energy failure that occurs for the first few hours, and then there's sort of a latency phase before we see a secondary energy failure in these babies that have suffered from a hypoxic insult. So the creatine seems to be helping with ROS production or reducing the amount of ROS in the brain in the first 24 hours. This graph just shows that data presented slightly differently. In terms of the mechanisms behind this, they are potentially twofold, and we weren't able to get at that, but it is likely that the creatine buffering during that hypoxic period actually reduced the amount of ROS being produced. But then also we know that creatine does have some capacity to scavenge reactive oxygen species. So it might be a compilation of the two, reduced ROS and also some antioxidant capacity of the creatine, which is leading to this outcome. When we then looked at different metabolites within the brain and also in the fetal circulation, both before the hypoxic insult and then that 72 hours after, we saw in summary that the higher the creatine levels, the lower the percentage change in partial pressure of oxygen and oxygen saturation in the systemic circulation of the fetus after the hypoxic insult. And we also saw a reduction in cerebral pyruvate and lactate accumulation after the hypoxic insult in our animals that had the higher levels of creatine. So this data is plotted slightly differently. It's not a group-by-group comparison because of that phenomenon that we identified where we saw the hypoxic fetuses have a natural adaptation for increasing their creatine concentrations in certain regions of the brain. So this data is looking at the area under the curve or the correlation between the level of creatine within the tissue and those metabolites. And then the final bit of data from these studies I would like to present to you today has only just been published in Annals in Neurology. And Nee and our team were focused on, after that secondary stage of energy failure, the presentation of seizures, which is a real challenge and red flag for our clinical colleagues in the NICU. Seizures are really hard to manage. We have to use really nasty drugs to manage them, and it's also indicative of longer-term neurological risk. So it's an important marker to focus on. And with colleagues at the Ritchie Centre that really this kind of neurophysiology is their bread and butter, we were able to look at seizure or electrographic seizures in these animals. So a seizure is defined as slow cyclic waves lasting for longer than 10 seconds with an amplitude of more than 20 volts. Anyway, that's just the detail. Here's the interesting thing over here. So this is the total number of seizures that was presented within our creatine supplemented UCOs versus our salines. So our salines, we had 6 of our 7 fetuses present with electrographic seizures. In our creatine supplemented animals, there were 2 of the 7 that presented with any form of electrographic seizure in that 72 hours after the hypoxic insult. Breaking down that data in a little bit more detail, we saw a statistically significant decrease in the burden of total seizures in our creatine supplemented animals. The number of seizures was almost reduced. And obviously, the time for seizure onset wasn't statistically different, but I'll show you some data in a moment, which is kind of interesting. Although I seem to have lost that slide. Anyway, so an interesting thing that we saw within the 2 animals from the creatine supplemented group that did present with electrographic seizures, we didn't identify them until longer than 25 hours into the insult, whereas the control animals started seizing within the first 3 to 4 hours. So again, some really interesting insights there into the potential energy buffering associated with creatine. So I just wanted to touch really quickly, I'm running out of time, on the safety of creatine. So this is a really important component of our studies. We're suggesting the potential of an antenatal supplement. If that was to be widespread, it would mean that a lot of fetuses that never experience any complications are going to be exposed to high levels of creatine during development. We need to make sure that it's safe. And so for the vast majority of our measurements, we see creatine doing absolutely nothing to the fetus in terms of their physiology, in terms of their outcomes. But from time to time, we have picked up a few minor things, so a few changes in gene expression levels, some changes in growth. And so whilst this isn't an area to be concerned about, it's just something to say that we need to continue to be diligent in generating this data, and just a call out to anyone who's doing these kind of experiments to make sure that we do that. So just really quickly for the back end of the talk, we know that creatine looks to be important, and it could be a potential treatment for hypoxic ischemic injury in the fetus. So now we need to know, is creatine tuned the same for all pregnancies? Is it tuned the same across a pregnancy? And who would potentially benefit the most from creatine? And so here we turn again to our prospective cohort study to get some answers. In all of our samples that we collected, so our blood and urines across pregnancy and then those samples collected at birth, we did some targeted metabolomics to look at both creatine but also GAA and the amino acids associated with or required for endogenous creatine synthesis. Today I'm just going to touch on some of the data from the creatine and GAA. Creatine levels are very stable across gestation in a human pregnancy. This is maternal plasma levels. But they are around 35% lower than what is published as being the standard kind of range in a non-pregnant female population. Our GAA levels do a bit of a swing, and so it is likely that this stability of creatine is in some ways being driven by changes in endogenous creatine synthesis across pregnancy. Really important finding from this study. In this healthy population where there were no complications, we saw no associations with creatine levels in the mum, in the placenta, in the cord bloods, and key measures of fetal growth. No associations. We did, however, see some characteristic changes though. So our male placentas of our male fetuses had more creatine, as did the level of creatine in the arterial cord circulation. So this is the fetal circulation returning back to the placenta. We saw creatine levels in the venous circulation increasing as gestation increased. And we also saw some maternal characteristics associated with decreased levels of creatine, where I'll just present really, really quickly some data from the vegetarians and vegans, because it's really important, especially given some of the beautiful work being done by Sergey and Darren, to suggest that pregnant women are likely not consuming enough creatine. And this is a case control study within the cohort looking at our non-meat eaters versus a control of meat eaters. We do indeed see a reduction in the level of maternal creatine in our non-meat eaters compared to our meat eaters in both their plasma samples and their urine samples across gestation. GAA samples, it wasn't statistically significant, but it actually seems like the GAA levels seem to be higher in our non-meat eaters compared to our meat eaters in the plasma. No change in the urine. So I popped this data in after our discussions yesterday about whether, our endogenous system can build up and replace any deficits that are coming from the diet. It seems like it tries, but at least in this population, it wasn't quite getting there. And this is just some measurements of creatine and GAA from our birth-collected samples that really reiterate that point. But given our numbers, we were likely underpowered to really be able to tease out what was happening there. And in this population, we did see some associations between maternal creatine concentrations and growth outcomes. So our newborn head circumference was positively associated with creatine levels, and also the length of the baby was positively associated with creatine levels, but just in our non-meat eaters. So finally, is dietary creatine a conditionally essential nutrient of creatine? Well, we definitely have data to suggest that it provides energy buffering and energy homeostasis to key tissues during pregnancy, and that a diet devoid of creatine lowers maternal creatine concentrations and at least statistically changes fetal growth. But I stress that these babies were all very healthy, good weights, good lengths, but there is a difference at a statistical level between our meat eaters and non-meat eaters. And could we use dietary creatine supplements to improve outcomes for babies, particularly those that experience hypoxia? Our preclinical models are suggesting yes. It's definitely not a panacea or a silver bullet, but it's helping around some key measures and outcomes. And I think any parents in the room, if they have gone through those horrible events, they'll take any potential benefits. And so this is the avenue that we're going to continue to pursue with our clinical colleagues. Thank you very much to everybody involved. A huge amount of work. I'm just here to represent everybody back at home, but particularly my mentors and dear friends, Professor David Walker and Professor Rod Snow, who took us and started us off on this journey. Thank you very much.